The title of this lesson is Movement of Molecules, and as we go through the presentation, you'll be using your graphic organizer that we went over in class. You will also be taking a look at Study Guide 2.3, and you'll be using your book to find the answers as we go along. So make sure you have all of those as well as your computer for today's lesson. We're starting with a review. So if you take a look in your book at page 56, up at the top there is a section called Before You Learned. In reading those three objectives, you will learn that cells have an outer coating called a cell membrane. Cells need starting materials, and cells must get rid of waste. These are all things that we have learned prior to this lesson. So what we're using that information for today is looking at how the starting materials and the waste go through the cell membrane. So we've talked a little bit about cells. The image you see here is of red blood cells going through a blood vessel. That blood vessel is also made of cells. So if you take a look carefully, you can see that there are things coming either into the bloodstream through the cells of the blood vessel, or perhaps they're going out of the bloodstream, again through the cells of the blood vessel. When you take a sheet of cells like that, and then you make them all connected and you can roll them into a tube, that is a perfect example of how tissue, which would be the blood vessel, can do something that individual cells can't do. You can't have individual cells form a tube like that, and so tissue becomes a little more specialized than an individual cell like the blood cells that are going through it. So these things that are going through the cell membranes of the tissue or the blood vessel, they could include gases like oxygen and carbon dioxide. They might include liquids like water, or they also might include solids like glucose and waste. But we think of gases, liquids, and solids as kind of big things we can see. However, all of these have to be in molecule form in order to actually go through the cell membrane. So this lesson discusses two types of transportation. First is passive transport. This includes diffusion, osmosis, and facilitated diffusion, which is obviously a special kind of diffusion. And then the other kind of transport is active transport, which includes endocytosis, exocytosis, and the sodium-potassium pump. So if you just think about the words passive and active, perhaps you can come up with the main difference between these two types of transport. Passive transport does not require energy, whereas active transport does require energy. That's the key difference between passive and active. And perhaps one way to help remember that is that active, doing activities, require your energy. So we're going to start our conversation with talking about the concentration gradient. It's really important to remember the concentration gradient because it's going to form one of our rules for either passive or active transport. When we talk about concentration, this means the number of items in any given area. So I want you to draw on your study guide in the upper right hand corner next to the vocabulary words. I want you to draw a slide. This is going to be our metaphor for concentration gradient. We're going to use this analogy quite a few times. So make sure you understand this. At the top of your slide, label it high concentration. And I want you to draw a lot of something on the top of the slide. Either a lot of people or a lot of circles or whatever you want to draw at the top of the slide. Draw lots. The top of the slide represents our high concentration area. At the bottom of the slide, I want you to label that low concentration and put just one item there. So after you've drawn this, now let's take a look at concentration. Concentration gradient simply means there's a difference between the top and the bottom. If you were sitting at the top of the slide and you basically just allowed gravity to do its thing, it would not require any energy for you to go down the slide. Going down the concentration gradient doesn't require any energy. Going up the slide, however, does require energy. So if you're here and you're going down the slide, no energy required. Climbing back up the slide, however, does require energy. So we either say that our movement of molecule processes go down the concentration gradient or they go up the concentration gradient. So which one of these two images shows a high concentration? If you take a look, both of the boxes are the same size, but there is clearly a different number of items in the box. Think about, what if you were one of these dots and this was an elevator that you were in? Which one has the higher concentration of people? It's correct, it's the one with more dots in it. If we change it just a little bit and look at, okay, let's pretend each dot represents a person, and now we have two different size elevators. If you count the dots, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, there's the same number of people in both elevators, but this one is a significantly smaller space. So which one shows the concentration at the lowest? It's again the one on the right because it has the most space. So concentration can be determined by the amount of space you have. So before we go any further, we've learned about concentration gradient. Now I want you to take a look at your graphic organizer, either the grid side or the Venn diagram side. Take a look at that and find the rule that deals with concentration. So if you're looking at that rule, it shows that both osmosis and diffusion, they go down the concentration gradient. So label that on your Venn diagram. Show that going down the slide. Active transport, on the other hand, goes up the slide or up the concentration gradient. So either draw pictures of slides with arrows going down or up, or use words in the appropriate places to label that rule so you remember what that means. All right, moving on. Before we can discuss the processes of osmosis, diffusion, and active transport, we need to know what a cell membrane looks like. We need to understand what cell membranes are. So I have a couple videos to show you that kind of describe what a cell membrane is made of and how things go through it as an overview. The cell membrane, remember, is made mostly of one of our 4 biomolecules. Let's see if you can remember which one.
Cell cytoplasm heading outside. Your last stop's a barrier 8 nanometers wide. The cell membrane, our subject for today, it's as basic for life as DNA. It's selectively permeable like the border patrol of a country. It maintains control, selecting what leaves or what gains entry. It's a guard bound to watch their patrolman a century, but not just a guard. It sends signals and nerves. White blood cells use their membranes to eat germs like hors d'oeuvres. Membranes are receptors, enzymes, junctions. So what kind of structure can have all these functions? C-double-L-M-B-R-A-N-E, control, transport, selective permeability, phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic by layer in us all. So let's take a look at what makes up the membrane. Phospholipids rule on this domain. In fact, membrane structure emerges directly from phospholipids' chemical properties. There's a head and a tail on every phospholipid. The tail's 2 long-chain fatty acids bound to a glycerol. It's made to order. The tail's non-polar hydrophobic, fierce water. The head's got a phosphate. It's charged negatively makes the head hydrophilic, plays in water happily. So tail avoids water while the head's attracted to it. When phospholipids form the membrane, that's how they do it. Cause when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Here the hydrophobic mode, water leave me alone. While the heads are sticking out, touching all those H2Os, tails in, heads out. It's how every membrane goes, tails in, heads out. In a cellular sphere, it's the bilayered bases and membranes everywhere. C-double-L-M-B-R-A-N-E, control, transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic bilayer in a solid. But a cell membrane's not just a phospholipid scene. There's cholesterol, carbohydrates, lots of proteins. In fact, membrane proteins have a presence so great they often exceed the phospholipids by weight. And all of these components are in constant motion, moving, mixing like a boiling potion, flowing like dancers in a party in Passaic. That's why the membrane thought of as a fluid mosaic. Let's start with the proteins since they're key in this mix. Transmembrane proteins span the entire width. Typically their ports think of channels of conveyance for things that don't go through the lipid bilayer. Any protein embedded in the hydrophobic middle built right into the inside is considered integral, while peripheral proteins either hang on the exterior or on the cytoplasmic interior. Cholesterol keeps membranes flowing with ease. In cold it keeps the lipids moving so they don't freeze. In heat it slows the lipids down, limiting the traveling. Cholesterol keeps membranes from unraveling. Membrane carbohydrates work as markers of sign, so your immune system knows which flag you're self-reliant. The blood types ABO and BNA are about the carbohydrates on the blood cell membrane. C-double-L-M-B-R-A-N-E, control, transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic bilayer in a solid. C-double-L-M-B-R-A-N-E, control, transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol, the fluid mosaic bilayer in a solid.
So in that video, you got a glance at what the cell membrane is made of. It has several structures in it, biomolecules that we've learned about before. The most numerous one are the lipids. Phospholipids is what the term the video used for those. Those are the most numerous things, but there's also proteins. Those are super important for the processes that we're studying, active transport, osmosis, and diffusion, as well as cholesterol. Cholesterol keeps the membrane fluid. It keeps it flowing. It makes it so it kind of doesn't get stuck in one specific spot. And some other things that we won't go into too much detail on. So here's another idea behind what the plasma membrane or the cell membrane looks like. Pay attention to this. You're going to be drawing cell membranes soon. You're also going to be looking at kind of an overview of how the various processes that we're studying work.
The plasma membrane is made up of several organic molecules. I will concentrate on two of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycel would form. A mycel is a sphere with the hydrophilic heads on the surface, the tails inside, away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins.
So as you saw in the animation just before this, you could see that the plasma membrane could move up and down, and this is why animal cells that lack that rigid cell wall can basically be any shape, and that shape then controls what they're able to do.
These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack. In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solute particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids.
So this is what we were talking about the other day with the with Bill Nye's the nucleus vibrating, that toy that was vibrating in the middle of the nucleus, or as the middle of the atom, causing all atoms to basically just bounce randomly all over the place, even in solid objects.
For instance, if a bottle of perfume is left open in a room, eventually, no matter where you are in that room, you are able to smell the perfume. The molecules of air hit the liquid, causing the liquid molecules to move into the air. At the time the bottle is first opened, there is a high concentration of perfume molecules in the bottle, a very low concentration of perfume in the air. As the perfume molecules are bombarded by the air, the concentration of perfume is dispersed into the air. In other words, the perfume is moving down a concentration gradient, high to low. This movement is called diffusion.
So diffusion goes down the slide. You can see there are lots of perfume molecules in the bottle and no perfume molecules in the air. So we're going from where there's a high to a low. That would be down the slide or down the concentration gradient.
Uses Brownian movement. Now let's take a look at another example of diffusion. In this aquarium of water, we drop some blue dye. As the water molecules move, they bump into the dye molecules. The dye molecules, at first in a high concentration, start moving and bumping into each other and the water molecules, moving into the area of lower concentration. As this movement continues, the dye spreads out and equilibrium is attained. Equilibrium is equal concentrations of the dye and water in every area of the aquarium. Remember, Brownian movement continues even after equilibrium is reached.
So this is where I want to pause this video for now, because we're going to learn more about diffusion before we learn about the other two types of transport. So again, let's take a look at our cell membrane. And I would like you to draw the cell membrane again on your study guide. Find a spot, maybe somewhere up near the review, and draw an image of the cell membrane. Here are some parts that you need to label. First part, as you're drawing all of your lipids, you should start with the head and the two tails. Draw a line to one or two of those and label those the lipids. Then, in the middle of your lipids, draw some proteins. Proteins go all the way through, and they act as tunnels or channels for molecules to go through the cell membrane. Make sure your lipids are in two layers and the tails go in the middle of the sandwich, the heads go on the outside of the sandwich. You're certainly welcome to draw the identifier proteins and cholesterol, which would be these yellow things, but the lipids and the proteins are the parts we need to focus on for the rest of our discussion on molecule transport. So in class, you are going to be acting out your diffusion, osmosis, and active transport as we get to each of these. So first, I want to start with diffusion. Remember, all molecules must stay silent. In your action scene, I want you to arrange yourselves into two highly concentrated groups. So, get up out of your seats, find your way to different areas of the room. Remember, all molecules must be silent and moving. Once you've found yourself in two highly concentrated groups, then you may diffuse into equilibrium. Show me what equilibrium looks like. All right, very good. Let's go over the rules for diffusion. If you take a look at your study guide, actually, let's back up for a second. In your book, find page 56. I'd like you to read the two paragraphs where it says, some molecules move by diffusion, and then answer question 2 after you've read those two paragraphs. Okay, now let's go over the rules for diffusion. Find your graphic organizer. Either side is fine, and make sure you understand these rules. First rule for diffusion states that molecules go from high to low concentration. If you have not done so yet, draw a slide next to this rule and show your arrow going down the slide. The next rule is that there's no energy required for this process. There's no energy required because if you go down a slide, it doesn't require any energy on your part. Gravity does the work for you. Diffusion moves only small molecules. These molecules can include, they're not the only ones, but for our purposes, what we need to understand is that oxygen and carbon dioxide are moving by the process of diffusion. And this process goes until equilibrium is reached. Remember, molecules never stop moving. So even once equilibrium is reached, if there is a large amount on one side and a small amount on the other side, eventually they equal out. But then, as they equal out, the molecules continue to switch sides because that's the nature of molecules. They just keep moving. So if they find yourself with more on one side, then some will go to the other side. This again goes down the slide. So let's take a look at some of the things that can affect diffusion. So we've already learned that concentration level affects diffusion. Let's learn one other thing that can affect diffusion.
Place 1 drop of food coloring into the petri dish full of water. This is to show diffusion in action. With diffusion, the particles of food coloring move from an area of higher concentration to an area of lower concentration over time. In the beaker to the left is hot tap water, and the beaker on the right is cold tap water. You can see how temperature affects the rate of diffusion. With higher temperatures, the rate of diffusion is higher, and lower temperatures, the rate of diffusion is lower.
Can you think of one more thing that affects the diffusion rate for different things, for molecules? So we've learned that the bigger the concentration gradient is, the more diffusion occurs. If I were to spray some Febreze in the classroom, if I were to spray more of that Febreze in the classroom, it would diffuse faster. If I were to heat up the classroom, it would diffuse faster, just like we saw in this demonstration. And there's one more factor that can affect the rate of diffusion. Think about if you took a spoon and put it into each of these glasses of water. So stirring or mixing up the molecules would also make the diffusion go faster. So now let's check your answer on number 2. Some materials move by diffusion. The question says, circle the diagram that shows the molecules after diffusion has taken place. The correct box should be this one. So diffusion, after diffusion has taken place, everything reaches equilibrium. On your study guide, please write the term equilibrium next to this box. That is spelled EQUILIBRIUM, equilibrium. All right, take a moment now on pages 57 through 59. Read the 3 red titles: concentration, diffusion in cells, and osmosis. And then fill in your word magnet on your study guide for number 3. Remember, you are trying to include the word osmosis somewhere in your answer. Now let's take a look at passive transport. You learned that a special form of passive transport allows polar substances to move between cells. Polar substances are things like water. Passive transport goes from high to low concentration. And the definition is the movement of materials through a membrane without any input of energy. Finally, on the right-hand side, your examples of passive transport include osmosis and diffusion. And something to remember is that passive transport is spontaneous. It just happens. There's no input of energy required. So take a moment now, remembering that all molecules are silent, and I would like to see 6 individuals arrange themselves as a cell membrane. So you're going to face each other with your arms sticking out towards each other. There should be 3 people on each side forming your double layer. And do this in an area of the room where there's enough space to walk around. Then we need 5 volunteers to represent oxygen molecules, and you're going to show us what oxygen does as it diffuses through the cell membrane. Go ahead, pick your people, and get started. Now that you've acted out diffusion, we're going to learn about the rules for osmosis. Find your graphic organizer and make sure you know these rules. First rule is that water molecules, it has to be water, goes from high to low concentration. So if you have not done so, draw a slide next to the concentration level for osmosis and point your arrow going down the slide. This does not require energy, and it also goes until equilibrium is reached. So this sounds like diffusion because it is diffusion. The only difference is that since water is such an essential molecule for all living things, it gets its own special name, and that is osmosis. So let's take a look at osmosis.
I put this yummy gummy through a science experiment that models what happens to a cell in an environment that's more watery than the cell is inside. And what I saw came as no surprise. My gummy expanded, quintupled in weight. My gummy expanded and increased so great. So listen up as Mr. W explains this. It's all about osmosis. Osmosis is the diffusion of water across a membrane or a water permeable border, diffusing like all molecules in fluid situations from higher to lower concentration. Osmosis, H2O diffusing. Osmosis, see the water moving. Hypotonic, the hypertonic flow. Osmosis, let's go. Let's talk about our gummy in osmotic terms. Hypotonic, hypertonic, isotonic are the words you can use to discuss water's watery diffusion. Learn them well to avoid confusion. Hypotonic means higher water concentration and relatively lower solute concentration. Hypertonic is the opposite, percent of water's less, with more solute dissolved inside, as you might guess. So let's take these terms and apply them to our gummy bear. I'm looking at the package and seeing what it says here. Its bear is mostly sugar with some other stuff mixed in, and holding it together is the protein gelatin. So when you put a gummy in a cup of H2O, it's readily apparent that the gummy is so hypertonic to the water that it's in, and the water's hypertonic to our little gummy friend. And so through osmosis, the water will diffuse into the gummy, which soon will look so huge. The mass it gains is water, which will infiltrate the gummy, which one day later is looking pretty funny. Osmosis, H2O diffusing. Osmosis, see the water moving. Hypotonic to hypertonic flow. Osmosis, let's go. Next I took this freshwater plant, named as Elodea. It lives in lakes and ponds, never in the salty sea. In freshwater, Elodea cells are full and firm. It's all about osmosis, as we're gonna confirm. See, the cells are hypertonic to their watery exterior, so water will diffuse into the cell's interior, expanding the membrane, pushing it against the wall. In fact, you can't see the membrane at all. But add some salty water, so the outside's hypertonic, and water leaves the hypotonic cells. It's so osmotic. The membrane leaves the wall, the cells inside the scrunch, with the chloroplast are stuck together in a bunch. That's why at the grocery store they always have that mister. The water on the veggies helps to keep them crisper. The droplets on the veggies are a hypotonic brew. Osmosis moves the water in the veggies look brand new. Dry fruit and beef jerky preservation is osmotic. The low water content makes the dry fruit hypertonic. So any germs that multiply upon them lose their water and die and don't contaminate the dry fruit in the water. Osmosis, H2O diffusing. Osmosis, see the water moving. Hypotonic to hypertonic flow. Osmosis, let's go. Animals, cells and situations hypertonic, lose their water, shrink and shrivel. Looks so sick, but an animal cell in hypotonic abode gains water and expands, eventually explode. Cause animal cells lack a wall, of course, so nothing pushes back at the osmotic force. Water flows from hypotonic to the hypertonic cell, which bursts cause the membrane can stop the swell. If you want to keep a frog hard outside a body beating, an isotonic fluid is what you'll be needing. Water concentration same on each side of the cell. No gain or loss of water, the cells feel so swell. And a paramecium, which lives in ponds and lakes, constantly fights osmotic water uptake. The contractile vacuoles of pumping adaptation to deal with this osmotic situation. Osmosis, H2O diffusing. Osmosis, see the water moving. Hypotonic to hypertonic flow. Osmosis, let's go.
So that gives you an idea of some of the reasons why osmosis is so important. As you looked at cells that could possibly burst if they can't handle any more water than what's normal. So it's important that anything with osmosis, anything acting out with osmosis, has an isotonic environment so that the same amount of water is both inside and outside. All right, it's your turn. Act it out. Remember, all molecules are silent. Again, we need 6 people to represent the cell membrane, and 5 people this time represent water molecules. So I want you moving back and forth through the cell membrane, showing osmosis. The last form of passive transport that we're studying is called facilitated diffusion. In facilitated diffusion, which you'll notice if you look on your graphic organizer, one molecule requires facilitated diffusion that we study. What is that molecule? That's right, glucose is the only molecule in our studies that require facilitated diffusion. So it works just like diffusion. Molecules go from high to low concentration, but this particular type of diffusion requires a carrier protein. There's no energy for this. The protein simply acts like a channel or a gate for various molecules, including glucose, to go through. Glucose doesn't need energy to get through. And you can see in the second animation here, the red circles represent glucose, and the black moving structure, that represents the carrier protein. So as the molecules go through, you can see they're still going from high concentration to low concentration through the membrane. All right, one more time. 6 people be the cell membrane. 2 of those people now need to figure out a way to act or look like a carrier protein in the cell membrane. So you're actually going to stand in the middle of that cell membrane and hold your arms out toward each other. And as you are the carrier protein, when the glucose molecules get close, one side of the carrier protein opens, and then the molecule goes through, and the other side opens to let it all the way through the cell membrane. Our last type of molecule transport is called active transport. As you look at this, it looks a lot like facilitated diffusion, but there's one big difference, and that is that little energy star that we see acting on that protein. So if you look at your graphic organizer, on the active transport spot, I want you to draw a slide where we're going from low concentration now up to high concentrations. We're going up the slide. This process uses energy to move substances. Cells use active transport to perform life functions. And an example of this is getting rid of sodium and waste. Your book goes over this when they talk about the marine iguana. So take a moment now, read page 59 with the title, actually 60 to 61, with the titles Active Transport, Endocytosis, and Exocytosis, and fill in number 4 with the following information. Take some time, now that you've read and filled in your study guide. Act out active transport. This is set up similar to facilitated diffusion. We need 6 people to be the cell membrane, 2 people again as that protein, and this time 1 person represents energy. 5 people in this action scene are going to represent sodium. So see if you can figure out how to act out active transport. Good. The last item, the last type of transport to discuss is endocytosis and exocytosis. And if you missed those two, I will back this up one more time. Take a look at the difference between endocytosis and exocytosis. So in endocytosis and exocytosis, you can see another picture of this in your textbook. In endocytosis, the cell membrane actually forms a bubble around any sort of item that needs to get into the cell and pulls that item into the cell. In exocytosis, it's the exact opposite. Think exo like exit, and there's a vesicle that's formed around whatever needs to get out of the cell, and that vesicle is made of the same lipids that your cell membrane has. When it gets to the cell membrane, it actually merges with the lipids of the cell membrane, allowing that molecule or other item to leave the cell. So you have one final question to answer on your study guide, number 5. You need to read the title that says Cell Size Affects Transport on page 62, and then fill in your final detail notes. When you are finished, this study guide may be turned in.